people developed, it's called GluCL ivermectin strategy, where people developed a tool that you can deliver either by injection or by viral strategy that hones in onto genetically targeted subpopulation of neurons. It activates a chloride conductance. So for as long as that channel is expressed in these cells, the neurons will be shunted. There's this large conductance, the neurons can't spike anymore. They're silenced. And this stuff naturally washes out after a day or two. So in other words, you have a tool called a silencing tool where I can say, well, if I can identify that this set of neurons is coded by those 3 genes, I can go to them and I can turn them off for 2 days. I can have a mouse that's raised perfectly normal, right? It's not a transgenic animal. It's a perfectly normal mouse, raised normally, and then for 3 days I can turn off. For example, I can turn off the neurons that I think are responsible for transmitting blue-yellow information. For example, in the condylocellular pathway from the retina to the LGN. And there are now these techniques that are becoming available. They have been widely available in invertebrates, particularly in the flies, but they're also now being developed for mammals. So we can silence. The early hope that individual genes will be very specific probably will not turn out to be true. If you look at the Allen Brain Atlas, by their counting, maybe less than 1% or closer to 0.1% of neurons have an incredible specific expression where they only express, let's say something like the claustrum. So probably we need techniques that can hone in on a combination of 2 or 3, maybe even 4 genes. Then we also have, for example, Ed Boyden at Stanford came up a couple of months ago with a fantastic technique where he took the channel rhodopsin 2 channels of the rod photoreceptor and incorporated it into a reporter gene. So essentially what he can do, he can include a photoreceptor into a specific neuron population. And now if you bombard that neuron population with light, you can turn on that, you can drive that neuron population. So let's say you have a smooth cortex, right, where you can put light on. Let's say you have a specific population, let's say that the one in layer 3 that project to MT, from V1 to MT, okay? Let's say you know that there's a specific gene that's expressed in them. Now what you do, you incorporate this rod photoreceptor, basically that's what it's related to that, into these neurons. So now they all have little photoreceptors. You put light on. All the other neurons, the other 99.5% of neurons, they don't care about it because they don't have this receptor in their membrane. But the 0.1% of neurons that happen to encode this, that all happen to be of the same genetic type, and they all project to MT, you can now drive them with light. You can essentially generate, it's like injecting current, but in each one of them. Right? It's like you had a microelectrode in each one of those neurons, except you can do it in the entire population. So now you can specifically drive just the population that goes from layer 3 in V1 to MT. So those techniques are incredibly exciting to me. Also what you want to do, you want to search, right now there are probably already a few thousand different strains of mice. And a lot of these mice may have already some very specific disturbances of consciousness. That, for example, that they're like zombie mice, that they can do delay conditioning but not trace conditioning, or that they can do all the behaviors that we can do without consciousness. And so you really want to do these random mutations like they do at, for example, Novartis Research Foundation, where they look for mutations that affect the blood pressure. But here I'm interested in looking for mutations that search for interference in consciousness. All right, so last slide. What is it we need? So we need robust animal models of consciousness. So we have them in vision using monkey, because there the similarity with monkeys and humans is always very strong. We need to develop in other animals, including possibly invertebrates like bees. We need to identify specific coalitions of neurons that give rise to specific NCCs. And then as I said, we need to inactivate them using all sorts of techniques. Direct electrical techniques, of course. We've been doing that since Penfield, right? The Penfield map that Ramachandran showed yesterday, well, we know that because you go in a patient, you stimulate with a microelectrode. But of course, that's very crude and that's invasive. There are also magnetic techniques that you can do today called TMS. Where essentially you have a magnetic field and you briefly switch on a magnetic field that induces current in the underlying neurons. You can use it in normal people, but it's very crude. It goes over square centimeters. That's why, again, I think ultimately we need these molecular techniques. And finally, what we need is really a principled. Everything I've talked about is sort of more an opportunistic approach, the one that Francis Crick and I've been taking, that we try to understand, we try to get hints from the neuroanatomy, we're looking at structures like the claustrum, etc. We try to get hints from experiment, what are the crucial elements, the crucial switching circuits necessary for consciousness. It's somewhat opportunistic. It's based on the available tools and techniques and insight into the brain. Ultimately, you need a principled theory of consciousness. We want to understand, under what condition is what Macintosh already conscious, but not telling me. And so what we really need is a principal theory of consciousness in the fullness of time. And the only candidate I see right now is the one that Jerry Edelman and Giulio Tononi are looking. I'm not sure this is the final thing, but the direction they're going in terms of information, integration, differentiation seems to be, to me at least, a very, very promising one. So with that, I'd like to conclude.
So, Christof, that was like a machine gun all the way through for an hour. But one thing that I didn't get is the mutations in the mice. So if I take this bat and I hit the mice on the head, it's going to go unconscious. And it goes unconscious for many reasons. Because I disrupt the sensory input, I disrupt thalamic processing, neocortical processing. I bet that you could have about 5 billion, 10 million, whatever, mutations that are going to affect, perhaps affect consciousness. I mean, for many different reasons, because they're affecting the sensory processing, because they're affecting any number of different pathways that may actually be supportive for consciousness or have actually nothing to do with consciousness. So I don't really know, and to take out layer 6, yeah, you're going to screw up a lot of processing. But does that mean that it's got anything to do with consciousness?
The idea is always to be, you're perfectly right, it can't be random, it has to be targeted. We're looking for things, for example, the animal can still do delay conditioning, but it won't anymore. So in other words, its auditory processing must still be intact, and its autonomic system must still be intact in order to have arousal, but this specific combination of storing information with the CS and then put it in the working memory, combine it 10 seconds later with the USZ, we're looking for that particular circuit. So it has to be very specific. And again, it's a strategy that's opportunistic. Like almost always in biology, we hope ultimately to interfere as specific as possible just with these 3 functions that require consciousness, not with all the other behaviors that do not require consciousness. I don't see how else we're going to do it in an experimental feasible way.
There's one question we got.
Okay. Nobody said it's going to be easy.
So you showed us an experiment of 2 yellow blocks disappearing. Is the experiment valid even with one eye closed?
Yeah. This one I showed you, yeah. So that's why I can show it here under these conditions. You can do it with one eye.
And it will still disappear.
This does not require binocular input.
Okay. The other question is, you're looking at neurons. It seems like if I equate it to a studying a computer and a black box approach, it's like I'm connecting oscilloscopes to different parts. I'm not studying the software aspect of it by just looking at signals. Is that an analogy? Or
no, I think it's a very pernicious one. I mean, that's the one I grew up with. That's the one we all grew up with. But there is no software in the brain, right? That just doesn't exist. There are neurons, there are voltages, there are dendritic potentials, there are synapses, but there's no software. There's no universal programming entity. There's no register of things like that. You can use those terms, but I think they're much more misleading, and they will lead you to construct totally erroneous theories.
Conditioning is one kind of programming, isn't it?
Yeah, you can think of it like that.
Okay.
You can think of it like that, but there's no universal programming. There's no programming language around. The program assumes a Turing machine, all those things. And I think those are all very misleading, and they don't really help you conceptualize the real problem of biology. That's my opinion.
Working? Yeah. So that was great, Christof, and I always find your talks very inspirational, and I agree with almost everything you say, but I always get stuck on certain things. So I don't want you to think I'm always like just trying to attack. But when you say on that slide that you accept the reductionist hierarchy and having, the fact that chemistry is based on biochemistry and so on, and then you start talking about computational primitives, it seems to me there's an inconsistency there. Because the point of the reductionist hierarchy is that there are not computational primitives. And I think we have to face in biology the very real possibility that it is not like the computer for exactly the reason that nobody's gone and built in some fundamental primitives. So can you either of the nervous system are, or remake the argument without mentioning the word computational primitive?